Okay, so our next presentation today, we'll hear from Derek Britton, Saskia De Vries, Michael Bice, Dan Bombadger, and Adam Blackert. This team will share how we can record activity in populations of neurons in awake animals while using visual stimuli to gain access to and to better understand information transfer and computations performed within biological neurocircuits. By mapping the neuronal activity within the circuits in the same animals, we hope to construct a framework on which we can build a model of the cortical computations underlying the sensory processing.
Hi, so yesterday you heard Christoph introduce the 3C framework of computation, components, and cognition. And this talk is going to concentrate on the axes of computation, components, in particular, how we connect these two things. So for the purposes of the next 25 minutes, what do I mean by these two things? For components, of course, there's the cell types, the morphologies, the genetic characterizations, but in particular, there's the connectivity between cells for a particular network. In addition, there's what that network actually does. What is its function? What's its computation? So here I'm showing a cartoon similar to what Stefan showed you, where this is supposed to indicate a kind of object recognition task. That image over there on the right is mostly statistically a grayscale image, but you actually have no problem noticing a tiger, and it pops out. You don't actually think about it. So this particular field, a subfield of connecting the network circuit architecture with the function, is called functional connectomics. So before we get started or dive into the data collection, I want to tell you about what theory has to say about the connection between these two concepts. So first of all, there's computation. In order to think about computation and its impact on architecture, you have to have some sort of hypothesis. And I've listed a set of hypotheses here, all of which we're coming up with theories and testing. Since I've got limited time, I want to dive into one in particular, and that's the idea of efficient coding. And efficient coding is something that's very attractive to a theorist because it basically takes the idea that the statistics of what you see in the natural world is very particular. So your monitor, for example, there are more configurations on your monitor than there are atoms in the known universe. So the number of images that you've actually seen is very small compared to the number of images that you could have seen. So in order for the nervous system to be efficient, the neural code actually has to adapt to this, or should adapt to this particular structure of images. So one of the nice successes of this theoretical framework is if you train a network on a set of natural images, you get a set of neural responses that look very much like V1 receptive fields. Now, the problem with this is if you assume one particular cell type, which is effectively what these kind of models do, there's an implication here. So if you have a couple of neurons that respond to very similar traits in the natural world, it's effectively redundant for these neurons to fire at the same time, so they tend to inhibit each other if you try to construct some architecture that implements this particular type of structure. So one of the things we're trying to do in the theory group is expand models of efficient coding to have multiple cell types. For first pass, I'm going to be happy with an extra cell type, let alone 42. 42 is a bit daunting. But then the idea is to see whether or not this is more biologically reasonable. On the other hand, we've got dynamics, we've got activity. The network has to do something. And the architecture actually has implications, for the information processing. And I've drawn a little cartoon to illustrate that. So say I'm measuring a couple of neurons' response to a couple of stimulus conditions. So here I've got my stimulus 1 and stimulus 2, which are a couple of types of gratings. And I have a scatter of points. Say that the red triangles to stimulus 1 and the blue dots to stimulus 2. In this particular case, the biological variability doesn't actually hurt me. And in fact, the correlated activity, notice the neurons tend to fire above average or below average in tandem, doesn't really hurt me. And in fact, I can draw a line between these two clouds of points telling me that no matter what's going on, as long as I measure both responses, I can distinguish the two states. This doesn't have to be the case. I could have had this particular pattern of variability, which means now the population response, if you will, confounds the two particular states. So there's a mathematical theory that we're working on, along with Eric Shea Brown, to use the mathematics of statistical mechanics to figure out the connection between a posited Wij matrix and the joint activity, the statistics of joint activity, and thus the information processing. We call this the theory of structure-driven activity. But in order to understand this, and link these theoretical concepts back to the 9 questions that we want to answer, we actually have to have a particular set of data. And in this case, the kind of data we need to have are the functional properties of a lot of cells measured at the same time. We need to know, so in this case, there's a measurement of calcium imaging where each of these cells has been labeled according to its feature preference, and we need that and many other things, models of the particular response of these neurons. And at the same time, we want detailed connectivity on the cellular level, which is what's depicted here on the right. So Saskia was originally supposed to put the func in functional connectomics, but you are in fact going to have to settle for just fun. So here she is.
Great. So so where I come into this project is to help us start to define the functional properties of the neurons. So this is a volume of the cortex, or the mouse primary visual cortex. You see the surface of the cortex is at the top, and we've imaged down through the different layers of the cortex. And there's a population of cells here that are expressing a fluorescent calcium indicator, GCAMP6, and we've expressed it only in a population that's found primarily in layer 4. So this is the input layer to the primary visual cortex. And we want to measure the functional properties, how these cells respond to different visual stimuli. To do this, we use 2-photon functional imaging. So using this, we can use the calcium indicator to record the activity of these cells in responses to visual stimuli. So this is just a schematic kind of outlining the details of the experiment. We've got a mouse. We've implanted a window in its skull so that we can image, using the microscope, we can image the calcium indicator. The mouse is awake and it's on a running disc. If it chooses to run, it's able to do so. Otherwise, it can also stand still. And then we present a variety of different visual stimuli to the mouse with a monitor in order to record the activity. I'm just going to mention right now that I did a lot of this data collection in collaboration with both Shomo Chatterjee in the neural coding group, and our RAs did a lot of work. Naveen and Brian did a lot of work for us for this, so I just want to call them out here. So during these experiments, we present a wide variety of different stimuli to the mice in order to characterize their visual responses that I'm showing here. We chose these stimuli so that we can capture both the spatial and the temporal response properties of the cells. So they include somewhat simplistic stimuli with gratings, sparse noise stimulus to map the spatial receptive field, as well as more naturalistic stimuli, including a battery of different natural images and a couple of different short movie segments that we show to the mice. We also include periods when there's no dynamic stimulus, there's just gray, when we can look at the spontaneous activity of the neurons. So while we present these different stimuli, we can then image the activity of the indicator through the microscope, and this is what we see. Now again, these cells are expressing a fluorescent calcium indicator, so whenever the cells are firing, you'll see this increase in the fluorescence of the cell. And so the question that we really want to answer here is, what are the features of the visual stimuli that are driving the activity of these cells? Or another way of putting it is, what is the visual features of, what are the things about the visual world that each cell is encoding? So by looking at the activity of each cell and comparing it to the stimuli that are being presented, we can now come up with a lot of different visual response properties. So I'm showing 4 example cells here. Each row is a distinct cell, and their responses to the different stimuli. And you can see that there's a wide range of different types of responses. I'm mapping here the spatial receptive field, primarily the off subunit for these cells. We've got direction and orientation tuning. Here's an orientation tuned cell. We show the response to temporal frequency. They're tuning for temporal frequency for the preference for the speed of the grading that's being presented, as well as spatial frequency tuning, as well as the responses to images. As I mentioned before, what I've done here is I've just sorted the responses to the different images, and we find that there's some cells that are highly selective for a small number of images. So here's an example where it has a very strong response to just a couple of the images in the set, whereas other cells, such as this cell here, has a large response to a wide range of the different images that we've included, and is less selective. And finally, on the far right, I've shown just a trial average response to the movie clip that we use. And what we've found is that for many of the cells, and these 3 here are good examples, they have a very robust and very reliable response. Every time we show the movie clip, we see the activity at the same time, where they're really responding to some feature in that movie. But the feature is different for these cells, as we see that their activity comes at different times during the clip. So you can see just from these 4 examples that we see a wide range of different types of responses from these neurons. And this is a very rich data set and will allow us to start probing a lot of the questions that Michael outlined about the correlations between these cells and their different functional properties. But ultimately, we want to know more than just what each cell does by itself. We really want to think about how the cells work together and how that relates to both their spatial arrangement and their connections. So here is the image that Michael just showed you a moment ago, where I've just looked at on one of these properties, so the direction preference, so which direction of grading motion the cell has its strongest response to. And we've just outlined each cell with the color of its preferred direction. So we can start to ask questions about if there's any sort of spatial structure here that we don't see much here in that regard, which is fine. That's what's been found in the literature. But at this point, we're simply looking at a single plane in the volume. This data is all collected at one single plane, and so we're only sampling from a small number of the cells in this volume. And to really look at the questions that we want to look at, we actually want to get as much data as we can from as many of the cells throughout this volume. So to do that, we return to this volume multiple times and image at different imaging depths on multiple days, and collecting the same set of, using the same stimuli, collecting that full data set at different imaging planes so that we have data from as many cells as possible. So on day 1, I can image from these cells. I can come back the next day, image from the cells above them. And again, on the 3rd day, I can get even more. And we can analyze these responses, and we can start to look at their properties throughout the volume rather than just in a single plane. And so now we can really start to use this data to start asking questions about the patterns of activity and the functional properties and the population, the responses of the population as a whole. So, what we find is that there's groups of cells that we see that are often active at the same time. So these are a group of cells, about 7 cells, that all have a significant response to this particular image of a bear romping in the snow. A different group of cells have strong responses to a different image of a bear, and a third ensemble of cells that are active in response to a kangaroo, I guess. And we can start to ask questions about these different ensembles of functional units. And we even find that there's cells that participate in multiple ensembles that are active at with different groups of cells at different times. And so ultimately, this raises the questions of how these functional ensembles relate to the anatomical connections that we find between these neurons.
Thank you, Saskia. So ultimately, as Saskia said, we see these ensembles of neurons, and we can utilize and visualize the activity of each of these neurons in awake behaving animals in order to map each of their kind of unique visual stimuli and visual responses to parts of the activity within the visual scene. And ultimately, as we see these correlated patterns of network activity across these neurons, but again, these are just correlations, and what we ultimately want to be able to understand is something about the kind of network behavior and functional connectivity between these neurons in order to constrain the models that Michael was talking about and really bring about some kind of framework in which we can understand these properties of computation and visual representation within the cortex. And really to get at that pattern of connectivity between neurons, one of the best ways that we can do that here is to look at the connections between individual neurons. And this has been shown previously and most elegantly, I think, by Forrest earlier, that in order to get at those patterns of connectivity, you really have to go down to this really fine scale to look at the individual connections between pre- and postsynaptic cells. And by doing serial section electron microscopy, we can then build out this volume in order to represent all of these neurons and map their connectivity patterns across this space of cortex that we recorded from physiologically. Now, in order to do this actually takes quite a bit of work. And I'm just going to talk about, before mapping this, how do we actually get that tissue that we recorded into a state where we can start mapping this connectivity using kind of high-resolution serial section electron microscopy. So we can look at the visual window. This is just a mapping of the surface vasculature from one of the animals that we recorded from, and these outlines in blue and red are representations of the primary visual cortex and then some of the higher visual areas. And as Saskia mentioned, we can target regions within that visual cortex for recording. In fact, this is about the size of that little window of recording that she showed the movie from with relationship to the rest of the brain. So the size of V1 is roughly 3 millimeters. We can then utilize this surface vasculature and also record all of the depth down through the cortical layers of all of this vasculature and all of the cells that you see responding within that volume. And what we really need is this kind of correspondence matching in the future in order to find these cells for serial section electron microscopy, because this vasculature pattern is what we can actually use as kind of a fiduciary marks and registration framework to kind of build out the correspondence between the optical physiology and ultimately the kind of volume rendering and serial section anatomical data. So once we get that mapping, we then take the brains out and we mark around this optical cranial window with fiduciary marks, and we can actually visualize the surface vasculature so that we can correspond these two particular maps onto each other and kind of have a good idea about, in the ex vivo brain, where that little region of interest we imaged from was. We can then cut up the brain using kind of block-faced imaging, similar to you saw before with IVSCC, and sequentially section through the brain and collect each of these sections as we're taking them off and record the images of the pattern of the cortex, as well as these individual marks to find ultimately this region of interest that we care about. And so we get that individual section out. We can then map at high resolution the pattern of the vasculature within that image, that individual cortical section in that depth profile, because ultimately we can confirm that our cells of interest that we recorded from are also within there. And ultimately we want to make sure that we're registering that volume, that vasculature pattern back to that kind of volume of vasculature pattern that we were able to capture from that original volume stack. And ultimately, the reason we are caring about this so much is that essentially we took this tiny little piece of cortex, tiny is relative, but it is quite small, and then we have to actually find all of those cells that we recorded from now in these really, really huge, quite high definition images. And this vascular pattern is really our key. It's kind of our guide to a needle in a haystack type of finding. Because really what you see is that those vasculature markers are visible in electron microscopy images, in the electron micrographs after the staining. And it's really that kind of registration framework that's going to allow us to go back in and demarcate all of those cells of interest that we saw and find them again and create a registration framework between the physiology and the anatomy. And with that, I'm going to turn things over to Dan Bumbarger, who's going to talk about some of the difficulties and ways we're trying to overcome them to collect this data.
I'll just use this. Okay, so once Adam has identified our region of interest, I am charged with taking this little block of tissue and cutting it into a lot of slices. So what I receive from him is an epoxy embedded piece of tissue that I can then start to cut up. And it's quite an extraordinary thing to do, because I have to cut these slices at about 40 nanometers. And just for a matter of perspective, if I were to cut up a human hair, that would be about 4,500 slices. And so they're very thin, and eventually we'll need to be counting our slices in the tens of thousands in order to get the size of data sets we need. And so it's quite an extraordinary undertaking. You can find plenty of references that refer to serial section data sets on the order of 100 sections being a large data set. And so we're operating in a realm that's very far outside what most people would consider normal for this kind of work. And so what I do is we have our very precisely identified region of interest in this block of tissue, and then I just kind of make a rough map of where I want to start to trim this block so I can stick it in the microtome, which is the cutting device, and I iteratively make some cuts, and then I check them, and by doing this kind of this iteration, I can get this define region of interest on my block with somewhere on the order of micron or 10 micron level of precision. And I trim the block by sticking in a microtome with a glass knife, and then just trim away the tissue until what I end up with is a little pedestal on the end of this little block. Little pedestal. In this case, this is a couple of millimeters by a millimeter wide, and so something a bit small. After that, I switch to a diamond knife. A diamond knife is very hard and it's very sharp, and with this I can cut the 40 nanometer sections. And what we do is we cut them, and the sections float off into a little boat of water. And as they're floating off, they form these ribbons, as you can see here. And
They're floating on water?
They're floating on water, yeah. And once we have ribbons, we can collect these ribbons onto these little copper supports that we call grids for historical reasons primarily. And these grids are covered with a very thin plastic support film that's transparent to both electrons and light. And this is the kind of traditional way of going about this kind of serial section. But one of the problems we're running into is that even the very best people lose some sections. And for us to have a successful data set, we can't lose consecutive sections. And so if we lose 2 sections in a row, that could potentially destroy our ability to recover information out of that data set. And just as a frame of reference, people that are pretty good at sectioning lose about 5% of sections. The people that are the very best lose on the order of 1% of sections. And that doesn't give us a very high probability of success, unfortunately, when we move into this space over here. And so, a very large data set would be in this space here, which is about, counting in thousands of sections, but we're going to be operating over here in the 25,000 sections. And so doing this by hand is probably not a very safe bet, even for the very best people at doing this. And so we're going to be moving towards automating most of what happens with our specimen handling in terms of the cutting. And our primary strategy for doing this is to essentially make a reel-to-reel system to collect the sections onto tape. And so this is what this reel-to-reel system looks like. I don't know if you can see the tape. It looks like an old cassette recorder, and that's essentially what it is. And there's a little nose on the end that actually sticks into this little boat of water to collect the sections. It's like a little conveyor belt. The tape itself, we use a laser to etch holes in the tape. And on those holes, we can put the same thin plastic support films we would normally put on these little copper grids. And this allows us to collect the sections in a relatively automated way, and also to do handling of the sections thousands at a time instead of having to individually handle each section multiple times, which is one of our major sources of loss. This is just showing this kind of tape collection device attached to the microtome, and just a little bit closer view here. You can see that it really is just kind of a little conveyor belt. So immediately as a section floats onto the water, it's kind of placed on this tape. And once we have this tape, it forces us to have to really reimagine the microscope. And so we can't just take a commercial transmission electron microscope and stick this tape in it. We have to substantially modify the column to both handle our substrates, which are quite a bit different from what most people use, and also to achieve the throughput we need to be able to image these very large volumes. And Derek is going to tell you a little bit about what we've done to modify our microscopes.
Sounds good to me. All right. Well, the first thing is, I suppose, to tell you what an electron microscope is, if you guys don't know what that is. So I'll do that really briefly. Transmission electron microscope, in this example, there's one right there, is a way in which we can accelerate electrons via electromagnetic optics through an ultra-thin sample, in this case, brain tissue, and then it's finally collected onto some sort of sensor. Traditionally, it was in motion film. We're going to be using CMOS sensors or some sort of other camera system. Here's a nice picture as well of the lab in the new building here of our 3 electron microscopes, and there's significant room for expansion in the future. But if you notice, have you ever been to a standard electron microscopy laboratory before? This is a relatively non-traditional setup, and I'll explain that in the near future. So the main question now is, how do we actually get high throughput with these electron microscopes? Traditionally, these types of microscopes are designed to load in manually 1 to 3, 4 sections at most. And if we're going to do 25,000 sections in series, that's going to be quite problematic. So with our collaborators at Harvard, Wei Li has designed grid tape. We're working now to bring that here and expand upon it as well. This is a method in which we basically modify the entire core or heart of the electron microscope, in which we can load thousands of sections in at one pass and have the entire system automatically collect these large electron micrographs. just to give you an example of why we actually need to use an electron microscope, the organelles that we're interested in are in the realm of, let's see if I can move this thing here, on the realm of about 10 to 50 nanometers. Visible light microscopy falls pretty short on that, and obviously we as people cannot see at those scales. So it's necessary for us to use a method like this, but we have to modify it significantly in order to get the throughput that we need. So here's a good cross-section of basically what we're designing here. They're called TEMCAs. This is a transmission electron microscope camera array for short, TEMCA. So you have your standard electron microscope here, the specimen that would normally be manually inserted in this area right here. And so we're going to take that out, essentially gutting that part of the microscope, inserting our own reel-to-reel system. We're also removing the traditional camera chamber, expanding the column down significantly, hence why in that picture of the EM lab space, you saw that those machines, if you notice, were on large stilts. And this allows us to have a magnified field of view and have, a large field of view and also have it magnified, in which we can use to then collect onto our camera array on the bottom. So this entire section here of the microscope is completely customized, as well as this. In addition to that, there's a significant amount of software that's been developed to automate the entire process. And then to give you a sense of scale of the data set sizes we're talking about, in reference, this was something we recorded earlier this year over the summer. This is moving forward into next year. We're hoping to collect about 2,500 sections at about 100 by 100 microns. And then what you've heard multiple times is the cubic millimeter, 25,000 sections. And you can see how this scales up quite quickly to almost 3 petabyte data set. So there's significant work that needs to be done in terms of segmentation, alignment, and registration before we get some decent answers from this. But we are optimistic. But finally, as we get to use larger volumes, this will allow us to actually understand these ensembles of networks that Saskia and others today have talked about, to actually understand the network properties on these local circuits. And so our goal is to try to expand to make as larger volumes as possible to get to this cubic millimeter, to try to start asking some serious questions about functional connectomics. And with that, I am done. Thanks.
Okay, it's question time. So if you have a question, please raise your hand and we'll come to you with a microphone.
So can you predict which cells will be coactive in the movies from the other properties that you measure of the cells? Or is it a mystery given the other kinds of receptive field properties that you measure?
Yeah, that's a great question. So I haven't actually done that quite yet, but that's exactly the type of analysis that we want to be doing, is taking the spatial receptive fields, the tuning responses, and use that to predict their responses to the natural images and the natural movies and see how well those align, and the ways that we might have to tweak the models that we can create of these cells based on their spatial temporal receptive fields, based on the natural statistics. So yeah, that's.
For the receptive field measurements during behavior, how do you, or is this a problem, do you account for eye movements? Because I saw you had a camera, so does the mouse, it doesn't fixate, right?
No, so we track the eye, and so we have the data to look at the eye position during data acquisition. I've found in these data sets that there isn't a lot of eye movement, so I haven't done the analysis rigorously, but because the eyes aren't moving kind of very often, I don't expect that it'll have a large impact. But for something like the movies or the images, that's where we might see it, right? So that's where that would come in. But we do include that data, so that's part of the final data set. Yeah.
I'm wondering if that initial data set that is completed is large enough to actually ask, at least in a proof of concept way, any of the questions you want to ask, test any of the hypotheses before getting the massive data set, or is it just not large enough to capture enough neurons?
Michael, you want to?
The shorter answer is no. I mean, there is a spatial scale of kind of the ensembles you see to ask that kind of question. There's the spatial scale of the actual dendritic arbors, axonal arbors of each of the individual cells, which places some kind of limitation on how many types of connections you can observe, what is the probability of those connections within that small volume that we captured. Our hope was to be able to actually ascertain something about necessarily thalamic input coming into that, maybe looking at some of these local connections between some of the neurons within those, but not necessarily these large volumes, because really the ensembles, the size of these neurons, to capture kind of some network behavior in that sense, we are looking at orders much larger than what we already have attained.
There. Do you have plans to do volumetric imaging, like moving piezo or something, so you can do it on this single session?
Yeah, and actually some of the data that we collected for this was done using volumetric imaging, where you can kind of image down through the planes kind of at the same time. The trade-off there is that you lose a lot of the temporal resolution. And so if we want to know anything about kind of the temporal properties of the cells, we lose some of that information by doing the volumetric imaging. So it's we use it for we did collect some data that way, but my personal preference is not to use it for this, but there's an argument to be made that it can be more useful. So we do have some of that, yeah.
Okay, any more questions? Oh yeah, plenty.
Does that vascular registration really work? That seems perilous.
Adam?
The short answer again is yes, but yeah, I mean, you have not only do you have a lot of this vasculature, but I don't know how well you could actually see it in the movies, but you get a lot of the cells themselves, the nuclei, the cells that are responding, but even the neighboring cells, you can even see some shadowing within those cells. So by having all of that information in concordance with the actual vasculature itself, you really can start parsing together all of these patterns of connectivity. So yeah, at least. You use it all. Yeah, you have to use, you're going to start with a kind of rough approximation and start using each of those individual elements to start teasing things down closer and closer to kind of move apart the discrepancies.
Have you considered supplementing the experiments you're doing with single-cell optogenetic stimulation, which seems have you considered supplementing the experiments you're doing with single-cell optogenetic stimulation, which seems increasingly feasible now to take on?
Yeah, yes, basically. We have considered that. Not necessarily in the context of this specific project, but in the kind of the larger local circuits and CAM projects. There's work being moved in that direction to include that, yeah.
Related question is, so you see coordinated activities among the ensembles. The assumption is that they are preferentially connected to each other, but alternative hypothesis is that they receive common inputs. So how do you deal with that? I mean, if you don't find preferential connections among the cells, how do you... Have you thought about this problem? How do you deal with it?
Yeah, so there's a lot of reasons why they could be active at the same time that could have nothing to do with their actual connections, right? And so that's why, that's kind of why you need both pieces of data to really look at that. But yeah, it could be that they receive common input, that they're responding to a common feature in the image because of the spatial location of their receptive field, but that they don't actually speak directly with one another. And so that's why we need to have the connectivity data to complement the functional data in order to really get at those types of questions.
You're not trying to see if they do receive a common.
Yeah, I don't. We look at what the data has there, right? And
okay, another question over here.
Yeah, another thing that might affect correlations between neurons, we know affects correlations between neurons in V1 is neuromodulatory effects. Acetylcholine is one. So do you have any plan to address those kinds of things? It seems like the behavior is completely unrestrained, and so you don't really know what the behavioral state is of the animal.
Right. I mean, we have some information about the behavioral state in terms of, the pupil diameter of the eye gives us information about that. So we do have that. I mean, in these experiments, we're not looking at any neuromodulation on a kind of physiological level, just looking at kind of behaviorally. So we can parse the data into periods when the mouse is running or when they're based on the pupil diameter and look at the effects that we see there. Yeah.
Yeah, I was just struck, and maybe somebody can explain it to me, the tissue preparation and the cutting and the floating, both of the array tomography and the EM seems to be very similar, right? But you're not using each other techniques because they're sufficiently different, or?
I guess, in essence, they are essentially, the cutting is essentially the same, right? They use the same microtome, they use the same diamond knife, they float the sections onto water. We're going to be moving towards sections where we're not collecting ribbons, but that's kind of beside the point. But they're mounting things on glass for visualization with scanning electron microscopy rather than transmission electron microscopy. And and they can do some additional things because of that, because they're very robustly mounted. They can do some staining for different molecules, which is really nice, so they'll have some additional contextual information. But our game is geared more towards scale and throughput. And so we will be able to process massive amounts of tissue, like on a scale that has not yet been done. And so that's, we're kind of going more for quantity with these kinds of data. But the cutting method itself is essentially the same. They are picked up differently and then handled differently after that.
Okay. I think that's probably it for questions. So thank you again, team.